All right, let's begin. Good afternoon, everyone, and welcome to the History of DNA Analysis. We'd like to start by introducing ourselves. My name is Malen, and we also have Alexis with us today. Before we get started, we wanted to go over some Zoom features. Your mic and video have been muted and turned off. You'll be able to use the chat feature, which is located at the bottom of your screen. If you select the chat icon, a box should pop up over to the side. If you have any questions, we ask that you type it into the chat, as we'll have a Q&A at the end of the presentation. In today's presentation, we're going to look at the history of DNA analysis, where it started and how far it has come. Before we get started, we would like to state that we are not experts. Rather, this presentation will serve as an introduction to the topic. Again, if you have any questions, we do ask that you hold it to the end of the presentation.
We wanted to begin the presentation with a basic overview of DNA. What is DNA? DNA, or deoxyribonucleic acid, is a molecule that is found inside human cells that contains a blueprint for your development and function. It is made up of 23 chromosome pairs for 46 total chromosomes that include the complete human genetic information that make you, you. Your DNA includes a roadmap for all of your genes, for your hair and eye color to your predisposition for certain diseases.
DNA is made up of molecules called nucleotides. These molecules include nitrogen bases, adenine, thymine, guanine, and cytosine. The order of these nitrogen bases in the DNA sequence form genes, which are the instructions for your DNA expression. All humans share 99.9% of DNA. However, it is the order of these bases that give us the variation between individual people. These genes are passed on from parent to offspring through their chromosomes. They contain the genetic history of your ancestors, and the variations are how we can determine the relationship between people.
There are 3 types of DNA that you can use to test your genetic lineage. Females have 2 X chromosomes, one from their mother and one from their father. Males have 1 X chromosome that they receive from their mother and 1 Y chromosome that they receive from their father. Mitochondrial DNA tests the DNA that is inherited from your maternal or mother's line. Only females pass on the mitochondrial DNA to their offspring. However, both male and female offspring receive this DNA and can test for it. X-DNA is DNA inherited on the X chromosome from your father. Only females have X-DNA. Y-DNA is DNA inherited from your paternal or father's line. Only males have Y-DNA. Exploring your DNA and genetic ancestry is often referred to as genetic genealogy. Genealogy is the study of one's family tree and ancestry. This method of tracking family history has been used most often. This is typically traced through archives and documentation throughout history. An example of this might be a birth certificate or a draft record from World War II. More recently, genetic genealogy has become accessible to the general population through at-home DNA tests. Genetic genealogy is a genealogy method that uses DNA testing to determine the genetic relationship between individuals by comparing their DNA sequences. The term genetic genealogy was first used in an article published in the Dallas Morning News on February 20th, 1989. This article, which was written by Tom Siegfried, explored the findings of a mitochondrial Eve. This mitochondrial Eve is the most recent shared ancestor for all modern humans. As previously mentioned, people often use DNA to find distant relatives or to fill in family trees. Scientists and historians also use DNA to determine genealogical relationships of historical figures. For example, you can track descendants from a founding father, or you can also use DNA to answer everyday questions about lineages, like finding the biological families of an adopted child. So, how did genetic genealogy and the use of DNA start? Let's find out. In this section of the presentation, we will look at how some of the early discoveries, advancements, and events have shaped the history of DNA analysis and led to direct-to-consumer DNA testing. Mind you, this coverage isn't extensive, but rather it contains a handful of advancements. DNA was discovered in 1869 by Swiss researcher Friedrich Miescher whilst he was studying lymphoid cells. In 1953, researchers James Watson and Francis Crick proposed the idea that DNA was organized in the double-stranded helix shape that we recognize today. In 1977, Dr. Friedrich Sanger, who some refer to as the father of genomics, pioneered a game-changing technique referred to as the Chain Termination Method, or commonly known as Sanger sequencing. While that might sound complicated, the strategies and methods existed to sequence protein chains. These strategies were problematic. When attempting to sequence nucleic acids, because DNA molecules are much longer. They're also made up of fewer units that are similar to one another, which make it harder to distinguish between them. The Sanger sequencing method made it so that scientists could read the specific molecular letters that made up the genetic code. By these letters, I'm referring to A, C, G, and T, which we mentioned earlier as being adenine, cytosine, guanine, and thymine. The Sanger sequencing method allowed scientists to read between 500 and 800 letters or bases at a time. This meant that DNA could be sequenced at a faster rate and more efficiently. The first complete genome to be sequenced using this method was the DNA sequence of a virus that contained over 5,000 nucleotides. Mitochondrial DNA, which contains over 16,000 letters, was also sequenced using the Sanger method. This DNA technique was pivotal in sequencing the entire human genome. In 1983, Dr. Kary Mullis developed a technique used to amplify DNA called the polymerase chain reaction, or PCR. The very basics of the PCR technique is that it takes a small sample of DNA, and in just a few hours, it creates larger quantities of that sample. This process is known as molecular photocopying, and it is considered to be rather fast. So why is PCR important? PCR technology is considered a fundamental tool in genetic and molecular biology research. The technique is used in several different ways. It's used in DNA sequencing, DNA fingerprinting, forensics, diagnosis of hereditary diseases, and detection of microorganisms. It's also a technique that is used in commercial DNA tests such as Ancestry or 23andMe. In 1990, an international effort was launched to map the complete human genome. A genome is an organism's complete set of deoxyribonucleic acid, or DNA. DNA molecules are comprised of two twisting paired strands. As mentioned earlier, each of these strands is made up of nucleotide bases adenine, thymine, guanine, and cytosine. There are about 3 billion of these twisting paired strands in the human genome, and the Human Genome Project set out to 1. Sequence the exact order of the nucleotide base pairs in a segment of DNA, and 2. Map and identify all of the genes within the human DNA sequence. The Human Genome Project was completed in 2003, 2 years ahead of schedule, and cost about $2.7 billion. This project, which sequenced 99% of the human genome, was sequenced to an accuracy of 99.99%. The project identified more than 3 million human genetic variations called nucleotide polymorphisms, also referred to as SNPs, among other accomplishments.
Next, we'd like to shift your focus to law enforcement and DNA. In this section of the presentation, we'll look at some of the discoveries and events that have shaped the history of DNA analysis as it relates to law enforcement. Before commercial DNA was readily available, law enforcement and private agencies used it to help solve crimes and unknown lineages. Initially, DNA was not a strong enough proof of guilt in criminal cases, as the technology to study it was too new. However, the 80s ushered in a monumental breakthrough. Alec Jeffreys, a British geneticist, discovered the genetic fingerprint in 1984. This development was made when Jeffreys was attempting to trace genetic markers through families, specifically looking for patterns of inherited disease-causing mutations in the DNA. While reviewing X-ray film image of samples, Jeffreys noticed specific patterns, and these patterns were distinguishable and they're unique. Essentially, with the exception of identical siblings, every person has a unique DNA fingerprint profile. Immediately, Jeffreys had ideas of how this discovery could be used, such as in paternity cases, crime scenes, and immigration cases where nationality and paternity are intertwined. At the time, it was unclear how stable DNA was, so Jeffreys conducted an experiment where he turned his lab into a crime scene. Over the span of 2 days, Jeffreys cut himself, leaving blood marks all around his lab. He then tested his bloodstains and determined that his DNA was intact. The discovery of DNA fingerprinting has had a huge impact on law enforcement and forensics, giving way to DNA profiling. The first use of DNA fingerprint and DNA profiling was in a case involving the rape and murder of 2 teenage girls in Leicestershire, England, between the years of 1983 and 1986. The police had a suspect in custody who confessed to only one of the killings. And using DNA fingerprinting, the forensic evidence showed that the 2 girls had been raped and killed by the same man. Interestingly, the DNA profile of the young man in custody didn't match the DNA profile left behind at the crime scenes. It was determined that the suspect in custody had provided false confession that was in part influenced by his mental disability. In 1987, police held a DNA dragnet asking men within the community to submit their DNA. There were over 5,000 men who willingly provided a sample of their DNA with the hopes of catching the killer. At the time, Colin Pitchfork hadn't provided a sample. However, he was overheard talking about a scheme where he would provide a DNA sample using his friend's name. The person who overheard this conversation contacted police, who then took him in for questioning. And while in custody, the police took a sample of Pitchfork's DNA, and it turns out his DNA matched the forensic DNA profile. So Pitchfork ended up confessing to the murders and was sentenced to life in prison. The first use of DNA fingerprinting in America involved a serial rapist who sexually assaulted 23 women in Florida. At the time, the identity of the assailant was unknown, but in 1987, a woman called police to report a prowler in her area. This led to the arrest of Tommy Lee Andrews. While in custody, the police were able to match his DNA as well as fingerprints to those left behind at a crime scene. His first victim was also able to identify him as her attacker. And in November 1987, Tommy Lee Andrews became the first person in America to be convicted based on DNA evidence, and he was sentenced to a total of 100 years in prison. Most recently, in 2018, forensic DNA profiling led to the arrest of one of America's most notorious serial rapists and killer, the Golden State Killer, otherwise known as Joseph DeAngelo, was arrested for crimes of rape, murder, and burglary committed in the 70s and 80s. The FBI sequenced forensic samples that had been frozen for decades. And while it was initially believed that law enforcement officials were able to apprehend the Golden Gate Killer using Ged Match. That wasn't the entire truth. For those unfamiliar with Ged Match, it's a service where you can upload your raw genetic data to its databases. Once you've completed a DNA test, you can use the Ged Match database to compare your genetic sequences with other people to see if there are genetic connections and relationships. And while Ged Match was used, it wasn't the first course of action. The FBI actually sent samples to FamilyTree DNA, but that didn't get any results, and there weren't any closely related matches. There were only distant relationships. So the DNA profile was then sent to MyHeritage, and that's where they found a close relative. From there, the FBI sequenced discarded DNA samples from the suspect, which matched that from the crime scene, leading to the arrest of the Golden State Killer. This case highlighted several concerns regarding DNA privacy issues and the use of commercial DNA databases by law enforcement. Several commercial DNA companies amended their policies to safeguard their customers as a result.
Commercial DNA. In this section of the presentation, we'll look at the emergence of commercial DNA and highlight the rise of a handful of well-known companies. While popular now, commercial DNA companies often did not start out as DNA testing companies. Ancestry.com started out as a genealogy research site with access to scanned documents and records throughout history. They only started autosomal DNA testing in 2012, analyzing the human genome at 700,000 markers. DNA testing was meant to provide geographical information about ethnicity and answers lineage questions between close relatives.
FamilyTree DNA, which was created in 2000, was one of the first DNA testing companies to emerge. At the time, they were known for their Y chromosome and mitochondrial DNA tests. They use a single swab DNA test to establish relationships between individuals and to determine ancestry. About 2 million people have tested with FamilyTree DNA. 23andMe was founded in 2006 as a health research company. Unlike its competitors who offered Y-DNA and mitochondrial DNA tests, 23andMe was the first company to offer autosomal DNA testing. Its first personal genetics test cost about $999 and was on the market in November 2007. By 2011, their database had over 100,000 people. In 2012, there was a dramatic cut to their testing prices where consumers could purchase a test for just $99. In 2013, 23andMe was forced by the Food and Drug Administration to seize all health test sales. The FDA determined that they had failed to show enough evidence of their marketing claims that their genetic health tests could determine your risk for certain medical conditions. So all health-related reports had to go through the FDA for review process. However, that changed in 2015 when 23andMe was finally authorized by the FDA to sell their genetic health tests. As of now, 23andMe offers 2 tests, Ancestry and Health, each at different price points. Their health tests cost about $199 and covers over 40 plus carrier status reports. At the time of 23andMe's launching, the cost of sequencing a single human genome was around $300,000. And while the cost of sequencing has considerably decreased over the years, it isn't a financially accessible option for your average consumer. So instead of using sequencing technology, 23andMe uses a technology called genotyping. Genotyping works by reading or spot checking a specific parts of a gene for mutations linked to certain diseases instead of reading the entire gene. Here's an illustration that demonstrates the differences. Notice with genotyping, you have specific words that are in red with 2 highlighted letters. With sequencing, all the words are red along with specific highlighted letters. So with sequencing, it captures everything, whereas genotyping it's looking for the specifics. And genotyping is considered far more affordable than sequencing. Commercial DNA tests have come a long way. Over 12 million people have had their DNA analyzed, and this was around 2018, so that has increased. Ancestry has tested over 7 million people, leading the way in the direct-to-consumer genetic testing industry. In this section of the presentation, we will highlight some of the ways DNA has been used by scientists and historians to demonstrate genealogical relationships of historical figures and answering questions about lineages.
So we'd like to turn your attention to the Thomas Jefferson and Sally Hemings case study. Thomas Jefferson was a founding father and the third president of the United States. Sally Hemings was a slave that Thomas Jefferson owned. Throughout the centuries, there has been a significant amount of debate surrounding whether or not the 6 Hemings children were also the children of Thomas Jefferson. Some believed that the Hemings children were fathered by Thomas Jefferson's nephews, Samuel and Peter Carr, rather than Thomas Jefferson himself. Another claim that exists is that there was a child by the name of Thomas Woodson, who was believed to be the firstborn child of Sally Hemings and Thomas Jefferson. So in order to answer these claims, several studies were conducted on the known descendants of Thomas Jefferson, as well as the suspected Hemings Jefferson descendants. For these tests, the researchers conducted a series of tests using Y-DNA on the descendants of 3 different male lines. And in the picture here on this slide, we have Thomas Jefferson, Sally Hemings, and then the last picture there is going to be one of the children believed to be the offspring of Jefferson and Hemings. So on this slide, we have a family tree listed here, with Thomas Jefferson being that lower square here. And then in the above, it's showing the relationship between his wife and Sally Hemings, as well as their relationship to his father-in-law. So it is important to note that Thomas Jefferson's situation is very complex. John Wales, his father-in-law, was the father of Thomas Jefferson's wife, Martha, as well as being the father of Sally Hemings. So as a result, the children of Martha and Sally actually shared 3 out of their 4 grandparents, which complicated things with DNA testing, since usually it's not as common for them to share that many grandparents and therefore that many DNA segments. So none of Martha and Thomas Jefferson's sons survived, as you can see in the list of children on the left side of the screen here. So there's no DNA available for testing the direct male line, the known direct male line from Thomas Jefferson. However, there were sons shown from the Sally Hemings, Thomas Jefferson offspring. So for these tests, they chose to test other relatives in Thomas Jefferson's family to determine whether or not he was the father. So a study was conducted in 1998 by Dr. Eugene Foster to determine whether or not there's any validity to the Jefferson Hemings question. And using that Y-DNA testing, which, as we mentioned earlier, is only available for males in a line, 19 genetic markers were tested on that Y chromosome of 14 different individuals. And it was analyzed and compared to who they believed was the descendants of these possible children. So those who participated were the descendants of people who were believed to be the father of Sally Hemings' children. So the male descendants of the people with asterisks on this chart. So we have Field Jefferson, who is the uncle of Thomas Jefferson. And then we have the two nephews down here, Peter and Samuel Carr. And then we have Eston Jefferson and Thomas Woodson. They were all tested, their descendants were all tested to see if they matched with the Y DNA of Thomas Jefferson. So the study found that the DNA from Thomas Woodson's descendants did not match Field Jefferson, which ruled out the claim that Thomas Woodson was a child of Thomas Jefferson. The study also concluded that the DNA of John Carr's descendants did not match that of Easton's descendants. So they were also not the parents of Sally Hemings' children. However, Dr. Foster's study found that the DNA of Easton's descendants matched that of Field Jefferson's, which shows that their Y chromosome DNA markers were identical. So what does that mean in layman's terms? It means that Eston Jefferson, the son of Sally Hemings, was also a son of Thomas Jefferson. So what that study took that to mean was that Sally Hemings and Thomas Jefferson did in fact have children together, and that her children were Thomas Jefferson's rather than another relative. While this conclusion validates that Eston was a Jefferson, without testing Thomas Jefferson's actual DNA, it is impossible to be 100% certain. However, the likelihood is very, very high, and the estate, Thomas Jefferson's estate, now recognizes those children as being his. So, based on historical records and accounts, travel patterns of Thomas Jefferson to his estate, there's a high probability that Thomas Jefferson is the father of Sally Hemings' children.
In a recent case, Delphine Boel, an artist in Belgium, used DNA to prove that she was the daughter of former Belgian King Albert II. It was a 7-year legal battle, but Delphine was added into the Belgian royal family, and she and her children were granted royal titles. Another interesting study is that of the Genghis Khan DNA study. In 2003, researchers found that Genghis Khan's DNA is present in about 60 million men alive today. The study found that the lineage originated in Mongolia approximately 1,000 years ago. Tests were conducted on the Y DNA line, or the male-only line of inheritance.
In many cultures, surnames are passed down from fathers to their children. As a result, many fathers and sons will have the same surname, making it easier to track Y DNA lineages. So these surname projects will often test males with the same surnames to determine when they shared a last common ancestor. As you can imagine, this is significantly easier with less common surnames, and significantly more challenging if you have a surname like Smith that is very, very common in countries. So these surname projects help to identify the general geographic areas where some of these surnames are more popular, which helps with ethnicity tracing. So for example, if you have the last name Smith, it's likely that you originated in England, and for example, Fritz is very, so things like that can also help pinpoint where your genetic origin might have been. DNA has come a long way since its initial discovery. There have been several technological and genetic advancements that have directly impacted the accessibility of commercial DNA testing. Advancements in technology such as that of genotyping, have led to efficient and inexpensive techniques for DNA testing. Breakthroughs in genomics, such as that of the Human Genome Project and DNA fingerprinting, have also been pivotal to the rise of commercial DNA and the amount of genetic information that is accessible to consumers for a reasonable price. So this concludes our presentation. We hope that you have enjoyed it. If you would like to contact us, you can do so by calling the Genealogy Center or filling out a Book a Librarian request. So right at this point, we can take questions if you guys have any. I believe that we had one earlier. Yes, we had one about how was the DNA of Thomas Jefferson obtained for testing. So if we're able to go back to that chart, I can explain where they got that. Give us 1 second. Yes. So for Thomas Jefferson's family tree, since the estate was not comfortable pulling DNA directly from Thomas Jefferson himself, they were able to test the DNA of descendants from people who were related to him. So up here in the top right corner, we can see the name Field Jefferson, who is listed as Thomas Jefferson's uncle. And since they are all descended from the same Y DNA, they were able to test Thomas or Field Jefferson, who would have the same exact Y DNA as Thomas Jefferson, because that's a carbon copy from father to son. So if it's following that Thomas Jefferson, the grandfather, to Peter Jefferson and Field Jefferson, and then going down to Thomas Jefferson, that Y DNA is going to be the exact same. So they were able to test the descendants, the living male descendants of Field Jefferson, to prove that Eston Jefferson was in fact the descendant of Thomas Jefferson. And since quite a few of those initial male line descendants did not have sons, it was Field Jefferson was the closest they could get to that. Were there any more questions? All right. So thank you for taking the time to attend our virtual program today. You can find more resources on our website, planolibrary.org, as well as our blog, planolibrarylearns.org. Oh, we had a question about testing female DNA to see if you are related to Abraham Lincoln's mother. So we have a advanced DNA class that we do on female DNA because mitochondrial DNA is a little bit more complicated. It's not a direct carbon copy from mother to daughter in the same way that Y DNA is a carbon copy from father to son. Mitochondrial DNA kind of merges in every reiteration of it. So whenever you have two X's, they combine to be passed on to it's a little bit more difficult to trace that back. You can find some areas that it will, but having that direct lineage tie is not as easy with mitochondrial DNA, just because it combines in different ways through every generation. And
if you're interested, oh, sorry.
Go ahead.
I was going to say, if you're interested in learning more about mitochondrial DNA, we have resources we can point you towards. And then we do have, as Alexis mentioned, some classes that we have taught in the past that are that have been recorded that you can watch as well.
And those are available on YouTube if you're interested That way, as with anything involving genetic genealogy, we always recommend that you have that paper trail before you start genetic testing, because it allows you to have kind of a starting location so you can know what surnames you're looking for. You can make sure that these people that you're connecting with, you know where they fall on your genetic tree and things like that. If you have more specific questions, you're always welcome to submit a book of librarian request through the Genealogy Center, and we can try and help you in the best way we can.
Are there any more questions? All right, well, thank you all for your time. We do appreciate it. You will be receiving a recording of this presentation within the next couple business days, so be on the lookout for that. And again, if you have any questions, please reach out to us via Book a Librarian. Thank you all. We hope you enjoy the rest of your day.